I will be talking about cross-experiment analysis, and here is what I will talk about today. First of all, I'll answer the relatively easy question, why do we need analysis across experiments at all? Then I will introduce metadata and how they streamline analysis. It's important to understand what metadata are and how we store them within Surtapetch software, and how we can review them as well, of course. Then I will introduce the data navigator and how we can use it to import Surtapetch experiments into a single experiment, usually you can call it meta experiment, and how we can reconfigure the data tree to efficiently extract a subset of experiments or subset of data that we want to analyze together. Then we want to see how we can analyze replicates. And finally, I'll summarize that, and then we'll switch to the combined Q&A session. So why do we do cross-experiment analysis at all? We want to analyze experiments that are performed in a replicate. We are all scientists. That means we do science. We don't do an experiment once. We do it somewhere between 3 times and 500 times. And we want to analyze that efficiently across multiple experiments, which have similar or identical structures. So we do the same thing multiple times. If we don't do the same thing, we don't have statistics. We don't have multiple samples of the same data point. And eventually we want to collect those results in a single file, single result file. Let me quickly talk about the data structure in Surtapetch software. That is mostly to understand the distinction between signal data and what we call metadata. What we define as an experiment would be the recording from either one cell or one preparation. Could be a tissue slice, could be a cell culture dish. And the routine, depending on what software you use to, you may be used to calling this either a protocol or a pulse generator sequence, is the collection of a family of steps, for example, like you could use for a sodium IV curve, one or multiple input signals, typically several sweeps. And the parameters around this routine, the metadata, are actually recorded into what we call a paradigm. We create a container for this routine. This is not a standalone data file. We record that into a paradigm, and if you repeat the same routine multiple times, the temporal context is retained, and also when exactly which routine was executed. So this will be an experiment that starts by executing a routine. An alternative approach would be what we call planned paradigms, where you can execute a paradigm that, for example, runs a sodium IV, then determines the maximum current, for example, to go on to find the half inactivation potential in a second routine, and then change to another paradigm that could be a dose response curve of a blocker, TTX, or whatever in our sodium example. And irrespectively of whether you use this approach, the planned paradigms, or the routine execution approach, what's interesting for the rest of the talk is what's in the paradigm. It's the metadata, and that's what we want to focus on. Metadata are stored either at the beginning of the paradigm, that could be hardware parameters, the computer, for example, what operating system am I running, what amplifier, what data acquisition am I running. It could be preparation details, what's the animal species, what genotype, or how was my sample prepared. And it could also be experiment descriptors like the test compound. Usually one experiment, one recording is one compound of interest that we're looking at. And then there's parameters that are stored during the experiment. For example, a solution exchange. We switch to a different solution, a new routine gets started, or the user adds a comment. All that gets stored to the metadata in the paradigm data. Metadata can either be automatically determined, like those hardware parameters. We know them, the software knows them, so no need for the user to enter them, or when a new routine gets started. Or they could be defined by the user, things like the preparation details, the experiment descriptors, or also, of course, the parameters like a comment or when a solution change happens. It's either manually tagged, or if the solution switch is controlled through software, it would be there. Now, how do we enter the metadata? You either go through the menu, Surtapetch 7 metadata, you hit Control and 9, or if you have a Mac, Command and 9, or you use the dashboard button That brings up the metadata dialog, and here you have a number of groups, metadata groups. Here are the parameters associated with the respective group, and in this relatively simple view, the animal identifier is something that you could define automatically. You could increment that automatically with each experiment. You can have a prefix, mouse in this example, and you can start at some value and increment that by 1 for each experiment. And you also may want to confirm the ID before each experiment. You don't usually switch your experimental animal halfway through the experiment. Another parameter you might want to add is the is the age. In this case, that would be 16. And note that it's a numerical value only because the units, in this case, the days, we could also use the P or E terminology, should be stored separately so that you can analyze a numerical value independently of its unit. You don't need to parse the whole thing. For the stimulus, in this case, we want to enter something, my test compound for one experiment, could be another test compound or a third test compound. That's the 3 test compounds that I used in the data that I generated for this presentation. If you don't find something in the metadata, a parameter that you would want to store, you may want to take a look at SERDA-Patch set preferences metadata, where you can increase the detail level. It comes in 3 detail levels. The default is basic. That's only a subset of all the parameters. And full exposes all metadata parameters. You have a number more groups, and within each group, you have many more parameters. So for example, if you wanted to keep track of the glass that you use because you suspect that there's a difference between in this case a 1.5 millimeter thick wall capillary and some other capillary, you can keep track of that as well. And if even in the full detail level there's a parameter that you think is of general interest, let us know. It's no problem to add new metadata parameters. At one point, you may get confused of what parameters you defined. You can go to Show Summary. You see a list of all the parameters that you did define, and by double-clicking on one of them, that takes you right back to the set metadata parameter. So that's about defining the parameters. Now about reviewing them. There's a variety of ways. One is in the Review scope, in the Scope window. You click on View Meta, and that opens the metadata review panel. And here is a tree structure that in this case shows you, yeah, here we have our mouse 16 days old, and here's our glass. And if you scroll down, the test compound in this case also is recorded at the routine level, assuming that the whole routine is under the influence of this particular test compound. One thing I want to mention is that we improved this appearance from what you have in the latest release version, 2.04. In 2.04, it's all white, and that makes it a little harder to read. So we improved the appearance. This will come in version 2.05. You can also review the metadata in the Data Navigator by clicking on the routine tree here and then clicking on the metadata link here. That will open a window similar to what you can access through the Scope window. Again, a tree structure, and here you have your individual parameters. Now the next step is combining experiments. If we have replicates of similar experiments like in this example, what we want to do is we want to open the Data Navigator, click Import, and navigate to the experiment that we want to import. For this example here, I have 3 experiments that I imported, and you end up with a tree that looks like this. The 3 experiments, obviously individual paradigms. There was only one paradigm per experiment, but how do we make sense of that? How do we sort that so that we can work with it? We click on Build Hierarchy and add the stimulus to the hierarchy. Select the stimulus group and add the compound name to the build hierarchy. And we put it all the way up top. And that way what happens is we have a reconfigured Data Navigator tree that has the test compound on top level, my test compound, another test compound, and a third test compound sorted alphabetically. That's why it's upside down. And then, of course, you can open individual tree branches, and here you see the 4 routines that I recorded into this experiment that I imported here. And normally what you want to do is you want to batch analyze series routine data that were recorded with the same routine. So what you want to do is you want to add the routine name to the hierarchy, and that up here, there were 2 routines that I used in this example. One is the sample routine bow tie test, the other one is the sample routine IV. And here in the IV, I get all routines that were recorded with IV, and in my test compound, I would probably want to analyze all these in one batch. What's important is, and this is one thing that almost tripped me up when I prepared this, if the series group, the meta parameter group, and along with that the parameter routine name isn't available, then you may need to increase the detail level. That's the slide I showed you earlier, the detail level of the metadata, because that's only available, I think, from the second level on. So next thing you want to do is you want to analyze the series, the routine data. You open it into the reanalysis scope window, you go to edit measurements, and then you select the last executed parameters. In this case, I made a modification of the original routine parameters from the sample routine IV that's in the sample routine pool. I modified it so that if I run the analysis, you only have the IV curve. By default, it would show you both an IV curve and a time course. The time course is not relevant in this case. And then what you can do is copy this to the layout by using the book scroll button there. And that creates a layout window that has the 2 signals and the results combined on one page. Then the next thing you want to do is add the analysis of another series. So you just run the analysis with the last executed settings, and that independently of what was done in that routine applies the last settings. And again, you add that to the layout, and you end up with the other 2 signals and the IV curve in the layout. And alternatively, what you can do is use the analysis editor, so the patch analysis editor through the main menu, and also add an analysis from there. Again, you click on the book scroll, and here this is identical to the second IV curve, and you can simply drag them on top of each other. And note that this was new in version 204. In version 203, you would have a white opaque background of the IV curve, so you wouldn't see the one that's below. This is a quick and dirty way of comparing 2 IV curves, but you can get as sophisticated as you like. So here, for example, I combined the 2 current traces. In this case here I had the capacitance compensation disabled. That gives me much greater capacitive transients, and that's what I measure in this model cell IV. In essence, I make the step voltage a little smaller, but you can also get as fancy as you like with editing these Igor graphs. This is in essence just by right click show this graph and then edit it. This is something that I edited in Igor itself, and that could be used for the publication already. Maybe without the shadow, some copy editors wouldn't like the shadow or some of that fancy stuff. So let me summarize what I told you today. Metadata enable the efficient analysis. They enable it by giving you parameters that you can sort your data by. Metadata have value in being recorded together with the signal data in one electronic file. It's not a paper journal that gets stripped of your electronic data at some point. You can use the data navigator to group series according to parameters you're interested in. You can combine replicate experiments into one. And I showed you how to use the analysis in the layout window to create publication quality figures.